Hello guys, welcome back to my channel. In this video, we'll be doing the second part of the molecular basis of inheritance, that is transcription and translation. In the previous video, we have already done the replication of DNA, and now we'll be doing this part and the last part of the lesson, that is the gene expression, which is very easy if you understand the transcription. So you all know that replication of DNA is very easy, and you all know about the enzymes involved and the mechanism of working. So now the transcription, that is also the same thing, and translation is slightly different thing, but you will easily get it after reading the notes. So let's get started without wasting our time, and before that, subscribe my channel and click on the bell icon. Molecular basis of inheritance part 2. So now we will first start with the transcription. Transcription is the process of copying the genetic material from one strand of DNA into the RNA. So that is actually a method of preparation of RNA from DNA using any one of the two strands of the DNA. This is called transcription. And the principle of principle of complementarity governs the transcription, except adenosine now forms pair with uracil instead of thymine, because we all know that in RNA instead of thymine we get uracil. So, and since adenine pairs with thymine, so in RNA now instead of thymine it will pair with uracil. And this is very important, that the principle of complementarity, it governs the transcription. In transcription, only a segment of DNA and only one of the two strands is copied into RNA. That's what I told you earlier, that it will not use both the strands, but only one of the two strands will be used. In a segment of DNA, only one of the two strands is copied into RNA. Otherwise, one segment of DNA would be coding for two different proteins, because what would happen is the one segment of DNA would be coding for two different proteins, and also two RNA molecules, if produced simultaneously, they would be complementary to each other, and hence they would form a double-stranded RNA, which would prevent translation, that is uh, formation of proteins from the RNA. This is translation, and it requires single-stranded RNA. If two different RNAs were produced, they would be complementary to each other, and they would form a double-stranded RNA. This would prevent the translation. So we'll go to the next slide now. Uh, what is transcription unit? A transcription unit in a DNA has actually three regions: a promoter, a structural gene, and the terminator. So actually it is a promoter and not a promoter. There should be a space between A and promoter. There is one promoter, the structural gene, and uh, a terminator. So I'll mark this with the yellow pen. So just see this one: a promoter, the structural gene, and a terminator. The DNA dependent RNA polymerase catalyzes the polymerization only in one direction. The DNA dependent RNA polymerase, it catalyzes polymerization only in one direction, that is 5' to 3', just like the DNA dependent DNA polymerase. And the strand which has polarity 3' to 5' acts as a template, that is this strand actually undergoes transcription, because the DNA dependent RNA polymerase has it does its activity from 5' to 3', and this strand which has 3' to 5', it acts as a template, and the other strand which does not it actually does not code for anything, it's called as a coding strand. The other strand with polarity 5' to 3' and a sequence same as RNA except thymine at place of uracil is referred to as coding strand, but this actually does not code for anything. So in this figure you can see a transcription unit. There is a promoter, structural gene, and a terminator. Promoter is located upstream with respect to coding strand. Upstream means towards the fifth end of the coding strand. This is a coding strand, and it is located upstream with respect to the coding strand, that is at the fifth end of the coding strand. And terminator is located at downstream, that is at the third end, 3' end of the coding strand. Okay, and this is the transcription start site. And you can see here that structural gene is between promoter and terminator. Transcription unit and the gene. Genes are located on the DNA, which is functional unit of inheritance. Genes are what? They are the functional unit of inheritance. And DNA sequences coding for tRNA or rRNA also define a gene. Cistron is defined as a segment of DNA coding for polypeptide. The structural gene is monocistronic, mostly in eukaryotes, or polycistronic, mostly in bacteria or prokaryotes. So the cistron, which is a segment that codes for the segment of DNA coding for a protein or polypeptide, is called cistron. And the structural gene is called monocistronic if it has only one cistron, and that is found in eukaryotes. And the structural gene is called polycistronic if it has more than one cistron. It is found in prokaryotes or the bacteria. In eukaryotes, the genes are split between the coding sequences or exons which appear in the mature RNA and the introns, which are actually the intervening sequences. These exons are actually helpful and we require exons, and that's why they are present even in the mature form. But introns are just like hurdles and we have to remove those introns. They are not useful, so they don't appear in the mature RNA. This is present only in eukaryotes, not in prokaryotes. These introns are present only in eukaryotes, and eukaryotes genes, that's why they are called as a split between coding sequences or exons and the non-coding introns. The split gene arrangement further complicates the definition of a gene in terms of DNA segment. Okay, actually a gene is nothing but a functional unit of inheritance, and the regulatory sequences are defined as regulatory genes, even though they do not code for any RNA or protein. They don't code for any RNA or protein. Just like the coding strand, it is a misnomer. The coding strand does not code for anything. It just acts, all the references are made with respect to the coding strand. That is, the promoter is located at upstream with respect to coding strand, and terminator is located with respect at a downstream with respect to coding strand. It does not code for anything. So similarly here, regulatory sequences are defined as a regulatory gene even though they do not code for any RNA or protein. Types of RNA and the process of transcription. So now in this part, types of RNA and the process of transcription. A single DNA dependent RNA polymerase catalyzes transcription of all three. Just a minute. Okay. So this a single DNA dependent RNA polymerase, it catalyzes the transcription of all three rRNAs, that is mRNA, all three RNAs, that is mRNA, rRNA and tRNA in bacteria or the prokaryotes. So in prokaryotes, for transcription there is only one enzyme, that is DNA dependent RNA polymerase, whereas in eukaryotes there are three different types of DNA dependent RNA polymerase. And in prokaryotes or the bacteria, all three RNAs, that is mRNA, rRNA and the tRNAs, they are catalyzed by the same or only one, that is a single DNA dependent RNA polymerase. RNA polymerase binds to promoter and initiates the transcription. It uses nucleoside triphosphates as substrate and polymerizes in a template dependent fashion. So this is very important, that this template dependent fashion is very important. It uses the nucleoside triphosphates as substrate and polymerizes in template dependent fashion following the rule of complementarity. It somehow also facilitates the opening of the helix and continuous elongation. So in replication of DNA, we have already seen that the opening of the helix is done by helicase enzyme, but whereas in this one, that is the transcription, DNA dependent RNA polymerase itself does the work of uncoiling or the opening of the helix, and then it continues the elongation process. Only a short stretch of RNA remains bound to the enzyme. Once the polymerase reaches the terminator region, the nascent RNA and the RNA polymerase falls off. This results in termination of transcription. RNA polymerase is only capable of catalyzing the elongation process. So there are basically three processes. One is initiation, elongation, and termination. Out of this, DNA dependent RNA polymerase, or simply you can say RNA polymerase, it does the work of only elongation, because for initiation and termination it requires some factors called initiator factor and terminating factor. Initiation factor is a sigma factor, and termination factor also called as a rho factor, to initiate and terminate the transcription respectively. In bacteria, mRNA does not require any processing, because we have already seen that in bacteria the RNA, they lack introns, they have only exons which are useful, but split chain arrangement, that is introns which are present between the exons, they are found only in the eukaryotes, that is monocystonic genes, and this is called split chain arrangement. But in bacteria or prokaryotes, they don't have such mRNA, so they don't require processing. And transcription and translation, they are coupled, because as there is no processing required, they take place almost simultaneously, and they are taking place in the same compartment. But in case of eukaryotes, as they have the well defined nucleus with a membrane bound, transcription takes place inside nucleus, but this translation takes place in the cytoplasm. So here you can see the transcription process. The 3 dash, there is here exons as well as introns present. They have represented this introns as a red part, and this is actually a eukaryotes, because here introns are present. But whereas in case of in case of bacteria or prokaryotes, they don't have introns. So these introns are removed by process called as a splicing. There are at least 3 DNA dependent RNA polymerases in the nucleus. In prokaryotes or bacteria, there is only one DNA dependent RNA polymerase, and that does the work of catalyzing transcription of all the RNAs, that is mRNA, rRNA, and tRNA, and also does the work of opening of the helix. But in case of eukaryotes, there are 3 different DNA dependent RNA polymerases. DNA dependent RNA polymerase 1 transcribes 28S, 18S, and 5.8S rRNA. DNA dependent RNA polymerase 2, it transcribes the heterogeneous nuclear rRNA, that is a precursor of mRNA, and DNA dependent RNA polymerase 3 transcribes 5S rRNA, tRNA, and snRNA. snRNA, that is small nuclear RNA. Now the primary transcript, that is the heterogeneous nuclear RNA, it is also called a split chain arrangement, because there are introns in addition to exons, and it is subjected to splicing, that is removal of introns. Introns are removed and exons are joined in a defined order to form a mature mRNA. Before maturation, it is called as heterogeneous nuclear RNA, also called as a precursor of mRNA. So we first remove introns using splicing. Splicing is called removal of introns, and then exons are joined in a defined order to form the mRNA. Heterogeneous nuclear RNA undergoes additional processing called capping and tailing to form the mRNA. In capping, at fifth end, and methyl guanosine triphosphate is added to the fifth end of the heterogeneous nuclear RNA, that is hnRNA. In tailing, around 200 to 300 adenylate residues are added to the third end of the template in a template independent manner, and this is required. The tailing is required to provide the stability. Fully processed RNA, heterogeneous nuclear RNA, is called mRNA. Okay, and that's why the hnRNA also called as the precursor of mRNA, and fully processed hnRNA is also called as a mRNA. It is transported out of the nucleus for the translation, because in eukaryotes and prokaryotes, both of them, translation takes place in cytoplasm. But in prokaryotes, both in eukaryotes and prokaryotes, transcription takes place, translation takes place in cytoplasm. But in eukaryotes, transcription takes place in nucleus, but in prokaryotes, transcription and translation both takes place in cytoplasm. So they don't require additional transportation of the mRNA. But in case of eukaryotes, transportation of mRNA is required after its maturation of hnRNA into mRNA. It is transported out of the nucleus into the cytoplasm where the translation occurs. And the split chain arrangement represents probably an ancient feature of the genome. Presence of introns is reminiscent of antiquity, and the process of splicing represents the dominance of RNA world. Okay, there are three additional processes required in eukaryotes for maturation of hnRNA. That is, first is splicing removal of introns, and then exons are joined in a defined manner. Then there is capping, in which methyl guanosine triphosphate is added to the fifth end of the hnRNA. And then in final step, that is the tailing, around 200 to 300 adenylate residues are added at the third end of hnRNA in a template independent manner. Genetic code. George Gamow suggested that the genetic code should be a triplet. Who suggested it? George Gamow. Triplet means it is a group of three. Okay, we say doublet when there are two, and triplet when there are three. So who suggested it? George Gamow suggested that the genetic code should be a triplet. Chemical method developed by Hargobind Khorana was instrumental in synthesizing RNA molecules with defined combination of bases. Marshall Nirenberg's cell-free system of protein synthesis finally helped the code to be deciphered. Severoka is a template. It is a polynucleotide phosphorylase enzyme which catalyzes RNA formation in a template independent manner. Enzymatic synthesis of RNA. What is Severoka? Severoka is a polynucleotide phosphorylase enzyme, and that catalyzes the transcription of RNA in a template independent manner. The polynucleotide phosphorylase, that is Severoka, was helpful in polymerizing the RNA with defined sequence इन अ टेम्पलेट इंडिपेंडेंट मैनर. सो एक्चुअली ये जो नीचे का पार्ट है वो एक्चुअली सेवेरोका के बाद आना चाहिए था. वो एक्चुअली टाइपिंग एरर है यहाँ पे. प्लीज़ कीप दैट इन माइंड एंड while taking down the notes, you write this first: Severoka enzyme, that is the polynucleotide phosphorylase, and then write the last two lines, and then again come to this point, that is template independent manner. Salient features of genetic code. The codon or the code is a triplet that we all know. That is, George Gamow suggested that a code should be a triplet. There are 64 altogether, 64 codons or codes. Out of that, 61 code for amino acids and 3 do not code for amino acids. So they are called stop codons or antisense codons. Sorry, they are called stop codons or nonsense codons. The code is degenerate, that is, some amino acids are coded by more than one codon, and code is highly specific. Each code codes for only one amino acid, and hence the code is specific and unambiguous. But some amino acids are coded by more than one codon, and hence the code is also called as a degenerate. And codon is read on mRNA in a contiguous fashion, that is without any commas or any other punctuations, hence it is called a comma-less. The code is nearly universal. Example, UUU code. It codes for phenylalanine from bacteria to humans. Exception, some variations have been found in mitochondrial codons and in some protozoans. AUG has dual function. AUG is a code. It has dual functions. Its first main major function is it acts as the initiator codon, and secondly, it also codes for methionine. It codes for methionine and it acts as a initiator codon. And the three stop codons or the nonsense codons are UAA, UAG, and the UGA. They are the stop terminator codons. Out of 64 codons, 61 code for amino acid and 3 are stop codons. Mutations and genetic code. Insertion or deletion of one or two bases, it changes the reading frame from the point of insertion or deletion and called frame shift mutation. This we have already you must have read about this in the previous chapter of the genetics, that is all those experiments done by Mendel's and all. In that last line, in the last part of the lesson, that is when we learn about the chromosomal aberrations and all, they have given this that insertion or deletion of one or two bases, it changes the reading frame because codon is a triplet. So when we add one or two bases, it changes the reading frame and it is called frame shift mutation. Insertion or deletion of three or its multiple in one multiple codon, hence one or multiple amino acids, and the reading frame remains unaltered. Because when we add three amino acids, we know that codon is made up of three different bases. That is, it is a triplet, and so when we add three bases, there is no change. So reading frame remains same, but when we add one or two bases or multiple of one or two, it does change the reading frame, and it's called frame shift mutation. tRNA as the adapter molecule. Here there is a printing mistake, that is, it should be a tRNA. Francis Crick postulated the presence of an adapter molecule that would read the code and bind to specific amino acid. The tRNA, then called as a sRNA, also called soluble RNA, was known before genetic code was postulated. tRNA has an anti-codon loop that has bases complementary to the code, and it also has an amino acid acceptor end to which it binds to amino acids. tRNAs are specific for each amino acid, and for initiation there is another specific tRNA called as the initiator tRNA. So there are no tRNAs for stop codons, and a secondary structure of tRNA, it looks like clover leaf, but the actual structure is like inverted L. Actual structure is inverted L, but the secondary structure of tRNA looks like clover leaf. Translation. Translation refers to the process of polymerization of amino acids to form a polypeptide. That is, we have seen the replication of DNA. Then after that we saw transcription, that is formation of RNA from DNA. Now we are looking at formation of proteins from the RNA, that is translation. It refers to process of polymerization of amino acids to form the polypeptide. The order and the sequence of amino acids are defined by the sequence of bases in the mRNA. The first phase, amino acids are activated using the ATP in the presence of ATP and are linked to the tRNA by process known as amino acylation of tRNA or the charging of tRNA. Protein synthesis takes place on the ribosomes. We all know that the ribosomes are called as a protein factories. It's because of this. First step is activation of amino acids in the presence of ATP, and then they are linked to that particular tRNAs, and this is called charging of tRNA or the amino acylation of tRNA. After that, protein synthesis takes place on the ribosomes. Okay, so ribosomes, they consist of structural RNAs and about 80 different proteins. It has two subunits. When a small subunit encounters the mRNA, the process of translation begins. So when the process of translation begins, when the small subunit of when small subunit of ribosome encounters mRNA, process of translation begins, and then the larger subunit binds to the mRNA. There are two sites in the large subunit for subsequent amino acids to bind and thus be close enough to each other for the formation of a peptide bond, so that protein is formed or a polypeptide is formed using the amino acids. And this formation of peptide bond is done by the catalase, that is 23S rRNA in bacteria and 28S rRNA in eukaryotes. Presence of catalase would enhance the rate of peptide bond formation. The ribosomes move from codon to codon along the mRNA. Amino acids are added one by one and they're translated into polypeptide, that is protein. What is translation? It is a polymerization of amino acids into polypeptide or the protein. The ribosomes move from codon to codon along the mRNA. Amino acids are added one by one because the tRNAs they join to the larger subunit. It has two sites for subsequent amino acids to bind. Actually, the tRNAs to bind, and tRNAs carry the amino acid, and they are so close that the formation of peptide bond is favored, and it is translated into a polypeptide. At the end of releasing factor, it binds to the stop codon, terminating translation and releasing the complete polypeptide from the ribosome. So this is the last step. At the end, a release factor it binds to the stop codon and terminating translation and the releasing completely polypeptide from the ribosome. Regulation of gene expression. Gene expression results in formation of polypeptide. It can be regulated at several levels in eukaryotes. The regulation could be exerted at transcriptional level, formation of primary transcript, split gene arrangement, then processing, that is removal of introns by splicing, and then there is capping, that is addition of 5-methyl guanosine triphosphate at the fifth end, and then tailing, that is 200 to 300 adenylate residues are added at the third end in template independent manner. Then there is the third step, that is transportation of mRNA out of the nucleus into cytoplasm, because in eukaryotes transcription takes place in nucleus, whereas the translation takes place in cytoplasm. And finally there is translational level, for that is polymerization of amino acids to form a polypeptide. Metabolic, physiological or the environmental condition regulate the expression of genes. So what are the factors that regulate the expression of genes? They are metabolic, physiological and environmental. Development and differentiation of embryo into adult organisms are also a result of coordinated regulation of expression of several set of genes. In prokaryotes, control of rate of transcriptional initiation is the predominant site for control of gene expression. Lac operon. Francis Jacob and Jacques Monod were the first to elucidate a transcriptionally regulated system, the Lac operon. Lac refers to lactose. Lactose is a sugar. It is a disaccharide. A polycystonic structural gene, that means which is found in prokaryotes or a bacteria. It is regulated by common promoter, a regulatory gene, and it's called operon. The Lac operon consists of a regulatory gene, then three structural genes, Lac Z, Lac Y, Lac A. I gene, I refers inhibitor. It codes for repressor. Z codes for beta galactosidase enzyme. Y codes for permease enzyme, and A codes for transacetylase enzyme. All three gene products in Lac operon are needed for the metabolism of lactose. Lactose is the substrate of beta galactosidase, and it regulates the switching on and off of the Lac operon. That is actually in the absence of lactose, the Lac operon is switched off because regulating gene the I gene, it forms the inhibitor, and inhibitor or the repressor, it binds to operator region and does not allow RNA polymerase to bind to the uh, to the operator region, to the promoter. So due to this it is switched off, but in the presence of lactose there is inactivation of the repressor, so DNA dependent RNA polymerase now binds to the promoter and the translation takes place. Transcription first takes place and then translation. So Lac Z codes for beta galactosidase, Lac Y permease, and Lac A transacetylase. Regulation of Lac operon by repressor is negative regulation. However, Lac operon is under control of positive regulation as well. It is also called as inducible because it can be induced by addition of lactose. Lactose does on and off switching of this Lac operon. Regulatory proteins can act as both positively activators and negatively repressors. Each operon has its specific operator and specific repressor. Human Genome Project, also called as HGP. This is very important, and a lot of questions can be asked from this part. It was launched in 1990, a 13-year-old project, and was coordinated by US Department of Energy and National Institute of Health and the Wellcome Trust. UK, Japan, France, Germany, China participated in it, and it was completed in 2003. Actually it was completed in 2006, and they had predicted about that uh, it would be completed in 2003, but unfortunately it was completed in 2006. It was a 13-year project and started in 1990, and that's why it is called as a mega project, also called as Human Genome Project or HGP. Human genome has approximately 3 into 10 raised to 9 base pairs, that is around 3 billion base pairs. Actually it is 10 raised to 9, but here the printing mistake has occurred, and the cost of sequencing in the beginning was US dollars 3 per base, so it comes out to be around 9 billion dollars, 9 billion US dollars, the price of this human genome project, because there are approximately 3 to 10 to 9 base pairs, that is around 3 billion, and cost of sequencing of one base pair is around 3 US dollars, so it becomes 9 US dollars, 9 billion US dollars, and that's why it is one of the reasons that it is called as a mega project. And HGP led to the rapid development of new area in biology called as a bioinformatics. This is very important. It led to a new field or new department, a new area in biology called bioinformatics. Many non-human model organisms like bacteria, yeasts, Caenorhabditis elegans, Drosophila, also called as a fruit fly, then plants like rice, Arabidopsis, they have been sequenced. So this might be asked that give the examples of the organisms who have been sequenced. So the examples are yeast, Caenorhabditis elegans, and Drosophila, bacteria, plants like rice, Arabidopsis, etc. Methodologies. Express sequence tags. There are actually two methodologies. One is express sequence tags. It focused on identifying all those genes that were expressed as RNA, and this is called as express sequence tags. It is highly specific. But there is another one, that is a sequence annotation. It is a blind approach of sequencing the whole genome, containing the coding as well as non-coding, needing vectors like BAC, that is bacterial artificial chromosome, then YAC, that is yeast artificial chromosomes. Okay, and the salient features of human genome projects are: human genome contains 3164.7 million base pairs, that is around 3.1647 billion base pairs. Every gene consists of around 3000 bases. Okay, and the largest gene is the dystrophin, which has around 2.4 million bases. The total genes estimated at around 30,000. Almost 99.9% of the genome is same in all the people, and functions are unknown for around over 50% of the genes discovered. Okay, almost 99.9% nucleotide bases are exactly same in all people, and the function of over 50% of the genome or genes discovered is unknown, and less than 2% of the genome codes for protein. Chromosome 1 has the most number of bases, there is 2968, and Y has fewest, that is 231. At 1.4 million locations, single base DNA differences, also called as SNPs or single nucleotide polymorphism or SNPs, occurs at 1.4 million. And SNPs can help in tracing the human history. So here there are many points to be remembered. One is human genome has around 3 point something into 10 raised to 9 base pairs, that is it comes out to be around 3 billion base pairs, and every gene consists of around 3000 bases, and the largest gene dystrophin has 2.4 million bases. Then 30,000 genes were discovered, and almost 99.9% genome is almost or exactly same in all the people, and uh, functions are unknown for over 50% of the genome discovered and or the genes discovered, and less than 2% of the genome codes for protein. Then the chromosome 1 has the most number of genes and chromosome Y is the fewest. And at 1.4 million places, single nucleotide polymorphism occurs. That is, a single base DNA differences occur, also called as SNPs or SNPs. These are few important points I have written here. Make note of this. Okay. So first one is the pressure of operon is synthesized all the time constitutively from the I gene. And that's why the I gene is called constitutive gene, because it is always expressed. And a very low level of expression of lac operon has to be present in the cell all the time. Otherwise, the lactose cannot enter the cells. Lactose or allolactose is the inducer of the lac operon. Hence, it is also called as a inducible, because by addition of lactose or allolactose you can switch it on. In the absence of lactose, it is switched off, and very small concentration quantity is required, because otherwise the lactose cannot enter the cell. Allelic sequence variation has traditionally been described as a DNA polymorphism. If more than one variant allele at a locus occurs in human population with a frequency greater than 0.01, DNA fingerprinting has much wider application in determining the populations and genetic diversities. Currently, many different probes are used to generate the DNA fingerprints. The hallmark of double-stranded helical structure of DNA is the hydrogen bonding between the bases from the opposite strands. And I didn't include the DNA fingerprinting part in this video because it's very easy, and I hope you can all do it your own. It's very easy. After reading you will get it, and if you have any doubt then please search videos on YouTube, because there are a lot of videos regarding this DNA fingerprinting, and hardly 1 to 2 questions might come from there, and it's very rare. So by reading you can easily understand it, and if you have any doubt then please comment me. I'll try to make a video on that. Okay, so thank you for watching my video.